So we have Chuck Gerba for our next presentation on zombie viruses and potential for waterborne disease transmission. And I'll mention while Chuck is getting set up that he was Kelly Reynolds' PhD supervisor. And so we're getting knowledge passed along here through multiple generations of students.
Is that working?
Yeah, that's right.
Okay, you just go.
Or you can use that.
Oh, okay. Thanks a lot for getting to talk to you today. I work at the West Center, which is a research center built by Pima County Wastewater to look at issues with water reuse in the future. Their idea was to invest in that now, figuring that in the next decades we're going to be going from converting wastewater to drinking water, which is actually already done in some parts of the United States. Give me a quick background. Treatment of sewage to drinking water, or toilet to tap, as some people call it. You must meet drinking water standards, which means you must reduce the probability of infection from viruses to less than 1 in 10,000 a year. That's infection, not disease, because it's too difficult to predict who's going to get ill. This requires no more than one virus in 100,000 liters of drinking water, which there is not technology to measure today, so it's done by removal. Removal was based on looking at enteroviruses like poliovirus, and current treatment used in California, Texas, Florida, is you have to have a 12-log removal to meet that based on what we knew about the concentration of viruses at the time. Now, California is proposing moving that to 15 to 20-log removal of viruses because of variability of treatment. If you're processing millions of gallons a day, there's variability in your treatment process. You can't consistently remove 6 logs all the time, they recognize. And so they did that. So previous knowledge is largely based on the enterovirus. This is polio, echo, Coxsackie, if you're familiar with it, because they're easy to measure in the laboratory, and using bacterial viruses standards. But our recent results, actually, of just 2 or 3 weeks ago, indicate these can't be achieved for the adenoviruses and reoviruses, which have not been studied very much for water and wastewater treatment until recent years. We did a study a couple of years ago, and it stood out the adenoviruses were one of the least removed group of viruses of the human viruses that we could find, which gave us a hint that we should probably be studying those a little bit more. I'll give you an idea. PPMV OV right there is pepper mild model virus. The viruses in the highest concentration in sewage are plant viruses. Pepper virus comes to all that salsa you're eating all the time. It passes through your gut, survives sewage treatment as an infectious. Interestingly enough, the plant viruses are pretty hardy. Some people speculated they cause stomach irritation, actually, an autoimmune disease. But so we have been doing a study at 5 different advanced wastewater treatment facilities designed to go from basically wastewater to drinking water quality. I'm just have time to show one of these results that you see here. Basically, it's a convoluted process involving taking wastewater from a sewage plant and then going through a series of adding chemicals, flocculation, ozone, chlorine, biological activated carbon, granular activated carbon, UV light, chlorinated again, and dump it into the groundwater for people to take up as a drinking water supply. This is designed to produce more than 18 log removal, exceeding what you're supposed to have for producing drinking water. I'm just going to give you the results from this plant here. Looking at the adenoviruses, we discovered that, yeah, you did pretty good at the first one. The first process with chlorination was designed to move 6 logs. The other ozonation was designed to move another 6 logs, only removed 2. It went down. The activated carbon in that, as you get further, you weren't removing anything. So the total for all this process, including pumping the water back up from the groundwater, only achieved about 6 or 7 logs, despite all these processes that were supposed to remove 6 logs or more of the virus. We also been looking at the bacteriophages because they're good indicators. We looked at somatic and male-specific bacteriophages, largely E. coli phages, Here we could see after UV light, you can get an 8 log or 8.43 log, similar to what we were seeing with the adenoviruses, just to give you an idea of what you're looking at technology. This is 4 viruses in 100 liters. My techniques for detecting viruses in water can detect less than 1 attogram in 1,000 liters. So that's 1 part per billion trillion by weight, by the way. So these viral methods are a billion times more sensitive than the chemical methods. And that's one of the reasons you can eliminate all the chemicals of concern and you still have viruses. Now, we looked at other processes too that integrate reverse osmosis membranes that are designed to desalinate water, take out all your organic chemicals, but they only give you a 2 log removal. Viruses are able to penetrate ultrafilters, our membrane filters fairly easily, actually, because when you look at the construction of these membrane filters, yeah, they remove 99.9% of the chemicals, but the viruses are in there, can penetrate these membranes, and they're only given credit for 2 logs, actually, membrane filtration processes. So removing the chemicals does not correlate with the viruses. Why are we seeing a large removal in the beginning and no viruses in the being removed to a high degree at the end? And that's largely because of the way people have been looking at it. The engineers, they assume you're always starting with this population here, chlorinating, and we know after about 4 to 6 logs, you get this, it levels off because you have a very resistant part of every population. But when engineers started designing treatment systems and go from sewage to drinking water, they assumed each process was starting here. In reality, after the first chlorination and ozonation, you're at the most resistant population there is. Somehow, there's always a resistant population. It's like, I think there's a big difference between a chemical. They're all the same molecules, right? But microorganisms are all like us. We're all a little bit different. And there are some real zingers here that seem to be incredibly resistant. I call this group that we've been looking at the zombie viruses because you can kill them and they can come back to life. Adenovirus, the one I just told you about, you can repair itself after UV light inactivation. He gave the example because it used to repair enzymes in your cells to repair the UV light damage. It also undergoes multiplicity of reactivation. That's when you take 2 inactivated viruses together and it hits the cell. One complements the other in terms of what areas are not inactivated in the genome on it. Adeno-associated are more the pendoviruses. They're born dead. They have to have another virus to infect them. The Boca virus, real viruses are the same thing, double-stranded RNA, most resistant viruses to chlorine disinfectant that we could find and naturally occurring. And there's different types that can complement and repair each other, as it turns out. That's what host repair enzymes look like. Basically, removing the cross-links that are established by UV light. In the terms of aggregates, it's known that viruses form aggregates, and my guess is that these are aggregated viruses we're looking at and making them so resistant. Just why real viruses are concerned. It's the most infectious virus we can find after chlorination and ozonation of any of the viruses we look at. Wide host range, the same real virus that infects humans infects cattle, infects bats. They cause a wide number of symptoms, but they're mostly asymptomatic, which makes it a little bit more difficult to really pinpoint. Been increasing episodes of them in bat reoviruses infecting humans. It's sort of like probing a population. If I go to try to find the next pandemic, they keep popping up in Asia as causes of different diseases all the time. So that's why the concern. Transmitted both respiratory and by water. It plays a role in a number of diseases. Adenoviruses, the one I've just been talking to, are of a lot of interest. Remember, there was a recent outbreak of children of hepatitis A, and that's been pinpointed to adenovirus 41 and the adeno-associated viruses. And somebody just published a paper less than a week ago that says, hey, when I looked at adenovirus and hepatitis and other new adenoviruses coming out, it seemed to be mostly occurring in areas that are doing wastewater reuse for drinking water. And so that caught attention. A lot of people have speculated the adenoviruses might be the next pandemic. And so where are we with this? With 1 minute left, is somehow we got to find out why these viruses are so resistant, develop new wastewater, drinking water treatment processes to do this. Already, hundreds of millions of dollars have been spent on these wastewater toilet to tap systems. in California just announced a $500 million system that won't work based on the data we've gotten in our recent studies. So it's a big concern. Basically, as biologists, we have to teach engineers about sex and explain to them that all microorganisms are not the same, because they seemingly made the assumption that all microbes are always the same, and they're not, based on the research. Obviously, there are toilet-to-tap facilities in the United States, and it's obviously they've been exposing people to adenoviruses all the time because nobody's been testing for them. Thank you.
So this is the first talk I've heard on sex and sewage treatment.
Hey, yeah, I was just curious actually which detection methods you use for these viruses that you mentioned were so sensitive.
Yeah, we're using cell culture for the adenos now. The trend in my field has been using PCR and molecular methods, but you can't tell live from dead. One of the reasons the adenos have been skipped over is because you have to do 2 cellular passages, use a certain cell line, and then do PCR to identify it. So it takes about 2 months to do an assay for the adenos, and most people don't want to bother.
If I'm allowed one other quick follow-up, maybe. How does this the virus titer compare in the wastewater treated to normal municipal water?
That's a good question. There have been drinking water outbreaks. It's mostly caused outbreaks in swimming pools of eye disease, red eye in swimming pools, but there have been drinking water outbreaks too. It is also, I didn't have time to say that, it's the most common virus in wastewater at the highest concentrations, because people develop chronic infections. They excrete it both in the feces and the urine. So one of the reasons we're probably finding it's in the highest concentrations all the time. You don't get seasonal variation with it.
Thank you very much for a very great talk. I never heard a wastewater talk this interesting before. And I have a kind of naive question in terms of the wastewater treatment, because you go through so many layers to reach the 8-log removal. However, do you feel with all the UV and all the treatment we have would actually stimulate some variation in terms of the mutation speed up for certain virus, because they are kind of being in a very high intensity, and you obviously going through a kind of a challenge environment where potential the virus left over may have the superpower to overcome the procedure. Then how do we handle that in the future when they're going back?
No, there's one of the things I brought to point of there is recent published, when they've all these treatment systems that evaluated chlorine, UV, they're doing a laboratory strain of virus they grew up in the lab. Recent published paper found out that you could find adenoviruses 10 times more resistant to UV light if you went to natural strains. See, most of this work is based on laboratory strains that's been done on wastewater treatment, not natural strains of the virus. But the mutation's a good point, too. One study published that if you keep viruses at higher temperatures in natural water, they become more resistant to chlorine. So there's been a concern that global warming may select for viruses more resistant to inactivation by disinfectants and UV light. Mm-hmm. Yeah.
So to me, this is very reminiscent of pesticides, where if you have a pesticide that kills the pest, of course it won't work very long. But if you have one that simply dissuades the pest from coming near, then that'll be more effective for longer because it's not a source of evolution. So in this case, are there any strategies at all for cleaning wastewater in which instead of trying to destroy the virus or the bacteria, you would rather be doing some sort of process to filter it out and avoid it?
Yeah, that's been tried. Like reverse osmosis membranes only give you about a 2-log because of the large area. The other areas to put it in aquifers and keep it underwater for a long time and travel. But you get that same phenomenon. We looked a lot on what they call soil aquifer treatment, and you get a big removal in the beginning and then very little. I mean, it goes out what's 3.8 kilometers. You can still find infectious viruses because you're selecting for the greatest surviving ones too. They survive longer. So filtration is difficult to calculate for viruses. Because one little hole, and when you treat millions of gallons, they go right through it. So it gets to be a real hard to eliminate them to that level from what we're finding out.
Thank you, Chuck.
Sure.